These are the diseases associated to macromolecules nucleic acid. So, the first one is the ovarian cancer. Ovarian cancer is when abnormal cells in the ovary begin to multiply out of control and form a tumor. So, if ovarian cancer is differentiated, the tumor can spread to other parts of the body. The next one is the Alzheimer's disease. Alzheimer's disease is a brain disorder that slowly destroys memory and thinking skills. Alzheimer's disease is very high at the age of 65. Down syndrome. Down syndrome affects a person's cognitive ability and physical growth. It causes mild to moderate developmental issues. So the next one is color blindness. Color blindness is when you are unable to see colors in a normal way.
Genetic code. The genetic code is the term we used for the way that the 4 bases of DNA, the A, C, G, and T's are strung together in a way that the cellular machinery, the ribosome, can read them and turn them into a protein. In the genetic code, each 3 nucleotides in a row count as a triplet and code for a single amino acid. So each sequence of the 3 codes for an amino acid and proteins are made up of sometimes hundreds of amino acids. So the code that would make one protein could have hundreds, sometimes even thousands of triplets contained in it.
Genetic code. DNA complementary base are G, C, A, G, G, T, A, G. But since it will undergo transcription for protein synthesis, it will decode into mRNA to form the original DNA C, G, T, C, C, A, T, T, C, to G, C, A, G, G, U, A, G. Therefore, in RNA, the thymine change into uracil. It also undergo translation wherein it repeatedly decode again to 3 RNA, transfer RNA. So it get the complementary base of mRNA to become or transfer tRNA, C, G, U, C, C, A, U, U, C. But in using codon chart to identify amino acid, it involves the series for protein synthesis. So group the mRNA into 3, C, G, A, G, G, U, A, G. For example, in using codon chart, we will identify the amino acids GCA. Let's find the G and C. GCA. GCA amino acid is alanine. For example, AG. AG, the amino acid is lysine.
DNA replication and RNA replication. DNA replication and RNA replication is a process by which DNA makes a copy of itself during cell division. Some viruses contain some enzymes required for replication. It contains RNA genome called reverse transcriptase. DNA replication takes place in 3 major steps. First is the opening of the double-stranded helical structure of DNA and separation of the strands. Second is the priming of the template strands. And lastly, assembly of the newly formed DNA segments. During the separation of DNA, the 2 strands uncoil at a specific site known as the origin. With the involvement of several enzymes and proteins, they prepare the strands for duplication. At the end of the process, DNA polymerase enzyme starts to organize the assembly of the new DNA strands. These are the general steps of DNA replication for all cells, but they may vary specifically, depending on the organism and cell type. Enzymes play a major role in DNA replication because they catalyze several important stages of the entire process. DNA replication is one of the most essential mechanisms of a cell's function, and therefore intensive research has been done to understand its processes. The mechanism of DNA replication is well understood in E. coli, which is also similar to that in eukaryotic cells. In E. coli, DNA replication is initiated at the auric-locus, to which DNA protein binds with while hydrolyzing of ATP takes place. DNA replication enzymes and proteins. DNA polymerase. DNA polymerases are enzymes used for the synthesis of DNA by adding nucleotide one by one to the growing DNA chain. The enzyme incorporates complementary amino acids to the template strand. DNA polymerase is found in both prokaryotic and eukaryotic cells. They both contain several different DNA polymerases responsible for different functions in DNA replication and DNA repair mechanism. DNA helicase enzyme. This is the enzyme that is involved in unwinding the double helical structure of DNA, allowing DNA replication to commence. It uses energy that is released during ATP hydrolysis to break the hydrogen bond between the DNA bases and separate the strands. This forms two replication forks on each separated strand, opening up in opposite directions. At each replication fork, the parental DNA strand must unwind, exposing new sections of single-stranded templates. The helicase enzyme accurately unwinds the strands while maintaining the topography on the DNA molecule. DNA primase enzyme. This is a type of RNA polymerase enzyme that is used to synthesize or generate RNA primers, which are short RNA molecules that act as templates for the initiation of DNA replication. DNA ligase enzyme. This is the enzyme that joins DNA fragments together by forming phosphodiester bonds between nucleotides. Exonuclease. These are a group of enzymes that remove nucleotide bases from the end of a DNA chain. Topoisomerase. This is the enzyme that solves the problem of the topological stress caused during unwinding. They cut one or both strands of the DNA, allowing the strand to move around each other to release tension before it rejoins the ends. And therefore, the enzyme catalyzes the reversible breakage it causes by joining the broken strands. Topoisomerase is also known as DNA gyrase in E. coli. Telomerase. This is an enzyme found in eukaryotic cells that adds a specific sequence of DNA to the telomeres of chromosomes after they divide, stabilizing the chromosomes over time.
The control of gene expression in the prokaryotes and eukaryotes. Let's first define what is gene expression. When we say gene expression, it is the process by which information from a gene is used in the synthesis of a functional gene product. This functional gene product is called proteins. What is the importance of gene regulation, or also called as the control of gene expression? It is to avoid wasting energy and raw materials in the synthesis of proteins, which the cell does not need or no longer needs. In prokaryotes, the control of gene expression is mostly at the transcriptional level. Transcriptional level, which is the process of DNA to RNA. Stated in here that regulation of transcription is the primary method to control what type of protein and how much protein is expressed in a prokaryotic cell. Written down below, we have the two arrow which states the situation wherein the resulting protein is no longer needed. If it is, then transcription stops. And if more protein is required, then more transcription occurs. Then the following subsequent steps occur automatically. On the other hand, we have the control of gene expression in the eukaryotes. As stated, it can occur at all stages of the process. Written down below are the stages or levels. First, the transcription level. It is when and how often a gene is transcribed. Second, we have the post-transcriptional level. In this level, the RNA is processed. But firstly, the selection of mRNA happens. Then it is transported to the cytoplasm. Next is the translational level. In this level, control happens by selection of which mRNA should be translated. Lastly, we have the post-translational level, wherein the protein is regulated by the following, either of the following: activation, reactivation, degradation, compartmentation, and modification.
Recombinant DNA is taking a piece of one DNA and combining it with another strand of DNA. The process of recombinant DNA involves combining the DNA of different organisms. The process depends on the ability